Two people can be born on exactly the same day, live for the same number of years, and yet reach middle life in remarkably different states of health. One may remain physically fit, cognitively sharp, and largely free of chronic disease. The other may already be experiencing metabolic dysfunction, declining fitness, or the early stages of several age related conditions. The chronological age are identical, but are they aging at the same speed? And if not, can we measure that difference early enough to change it? Welcome back to Decoding Longevity. I'm your host, Dr. Raghav Sehgal, alongside with my co-host, Dr. Macsue Jacques.
Hi, everyone. In our previous episode, we have explored how longevity interventions are tested and why rigorous clinical trials are essential before claiming a therapy slows aging. But that creates a fundamental challenge. Aging unfolds over decades, and we can't wait to 20 or 30 years sometimes to know whether the intervention is working or not. Instead, we need reliable biomarkers that tell us whether biology itself is changing long before disease develops. Today's guest has helped to build some of the most influential tools for doing exactly that. Professor Daniel Belsky is an associate professor of epidemiology at Columbia University's Mailman School of Public Health and faculty member of the Robert Butler Columbia Aging Center. His research focuses on understanding why people age at different rates and how genetics, lifestyle, and social environments shape health aging across the life course. He's one of the developers of the DunedinPACE, a DNA methylation biomarker designed to measure how quickly a person is aging, and now leads the ARPA-H FAST Initiative, one of the largest efforts to validate aging biomarkers for future clinical trials. Professor Belsky, welcome to Decoding Longevity.
Oh, very happy to be here.
Well, Dan, we've talked a lot, but I want to start with a very overarching question, which I think sort of, in some ways, is summarizes the start of your work. So many people are familiar with biological age, a blood test, or maybe a few blood tests that could tell them whether they're biologically younger or older than their chronological age. Your work focuses on something very different: the pace of aging itself. Could you explain the difference and why do we really sort of need to measure this pace of aging instead of biological age itself?
Well, thanks for asking, Raghav. This is definitely something we spend a lot of time trying to explain. I think that what I've settled on is the most straightforward way to explain this is a little bit like the instrument panel on your car's dashboard.
Mhm.
We can think about biological age as an odometer-like reading. It tells you where you are, how far you've traveled. But it doesn't tell you how fast you're going and therefore how you expect that location where you're observed, your biological age, to change going forward. The effort to measure pace of aging was designed with that limitation in mind. So what we're really interested in is how rapidly people's bodies are changing when we see them. So in that way, the pace of aging is more like the speedometer on your car's dashboard. It tells you the rate at which you're traveling and therefore is designed to be both more sensitive to changes that you might make to modulate biological aging, but also to give us a clearer sense of where you're headed into the future.
That is quite an incredible study. So the Dunedin PACE grew out of the remarkable Dunedin birth cohorts, where everyone was essentially, the same chronological age. And it's quite incredible that, the same people stayed in the study for so many years. But this became such a powerful design for measuring biological age. Could you explain why? And when you follow participants over decades, how early did you see meaningful differences in aging trajectories begin to emerge?
Yeah, so let me start with the first question about the Dunedin study and why it provided such a powerful platform for developing this novel aging biomarker. When we began this work, and the original study was published over a decade ago in 2015, aging was something that people thought happened late in the life course in humans. So human aging research focused on people who were in their 70s and 80s. And if you were a real pioneer and you were trying to expand your scope of what you imagined aging could include, you were looking at people who were maybe in their 60s or, for the exceptionally ambitious, perhaps the 50s. But this left out the entire young to middle adulthood segment of the life course, where in animal models, at least, we know that the molecular changes that are the root causes of aging-related disease and disability are already accumulating. And so the Dunedin study, in following a cohort of children from birth, which in this case happened between 1972 and 1973, across their lives, gave us an opportunity to understand how people's bodies were changing as they aged during this earlier part of the life course. The Dunedin study, like I said, enrolled its participants at birth, but they didn't begin conducting comprehensive assessments of their physiologies until they were 26 years old. So that formed the baseline in our study. And we then analyzed data originally extending forward from age 26 through age 38, and more recently, and with the development of DunedinPACE, through age 45. The study's back in the field at the moment, actually completing a collection of data for the half-century mark assessment, and there are plans to keep going. So the Dunedin study is exceptional in part for its capture of this earlier phase of the life course, but also, as I think you alluded to, its extraordinary retention of participants. And so one of the concerns we have in studying aging in human populations, particularly later in the life course, is that aging is a cause of disability and mortality, things that prevent people from participating in research studies. And what that means is you have some selection on your dependent variable in your study design. And this is helpfully avoided in the context of the Dunedin study, where they're starting much earlier, but also because they've managed to retain, through at least the age 45 assessment, 95% of the surviving members of the birth cohort. You have essentially no aging-related mortality by age 45. So what that means is that nearly everybody who was enrolled in this study has contributed data across the assessments. And that means we can make measurements of how fast people's bodies are changing without concern that the fastest changing bodies are disappearing from follow-up.
Yeah. And one of the things I remember, Dan, which you sort of, I remember this when you were presenting at the Biomarkers of Aging meeting, I think 2 years ago, which you pointed out, which was so unique about the Dunedin study, was also sort of, which is sort of missing in a lot of the other epigenetic talks, is sort of the cohort bias that exists, and that the absence of that in the Dunedin study, because it's the same sort of group of people that were born in the same year and followed through. While in a lot of the other cohorts, it's particularly challenging, or well, you're building these epigenetic clocks or other clocks in the cohort. It's particularly challenging to do that because they are across so many different generations, with each of them having their own biases. Isn't that true?
Yeah, so if I can pick up from I think what you're reminding me of here,
Yeah.
we talk about two really important sets of biases that may confound traditional aging clocks. And I spoke to the one that we, as epidemiologists, will sometimes refer to as survival bias or healthy participant bias, right? People are not observed because they've died or because they're not healthy enough to participate. But there is another set of biases that you're raising here that demographers call cohort effects. And here what we mean is that at a given point in time, a person's age is the same information as their year of birth. And when we have a mixed age population in our study, what we're observing are a mixture of birth years. And so just as we might want to infer the difference that occurs with aging by comparing a 20-year-old to a 70-year-old, we're not necessarily convinced that that same information is captured in the differences between births that occurred, let's say, in 1920 and 1970. And what changes over that historical period are many things, including exposures and healthcare interventions that may embed in a person's biology and not be distinguishable from aging processes in a cross section of data on people of different ages. And so for this reason, it's critically important when defining what aging is to follow the same people over time so we can watch aging happen to a person rather than trying to infer it from differences between people born at different points in history.
So the Dunedin cohort is, of course, so exciting and interesting, partly because also you have sort of this longitudinal measure that you have done, that you talked about, which I don't think is available in a lot of other cohorts except for probably the Baltimore longitudinal study of aging that I can think about. And that makes it really unique to model sort of these longitudinal changes. Partly, all of these clocks that came before DunedinPACE were either sort of trying to measure chronological age or mortality, but you were the first one to measure this longitudinal change or quantify this longitudinal change itself. Why was this shift so important?
Sure.
And does this kind of fundamentally change how we look at these aging biomarkers and what we're actually measuring?
I think that one of the things that people who do longitudinal studies, people, studies that follow the same people over time, recognize is that what they can see changing in individuals is sometimes not the same as what people infer from comparisons of people who are older and younger. And we talked already about survival bias and cohort effects that are a big part of that. We think that this longitudinal approach to measurement is critically important in the context of aging because many of the interventions that we envision to promote healthy longevity will be interventions that slow the pace of aging, but may not necessarily reverse it. And for this reason, the speedometer approach to measurement, the longitudinal rate of change, is potentially a more relevant quantity for evaluation than something like biological age, which is defined as the accumulated change across an entire lifespan. So if we think about the context of like a 2-year clinical trial, which would be a relatively long geroprotector trial or trial designed to target aging biology with the goal of modifying healthy lifespan, we're really not moving the needle a lot on the full lifespan of biological aging. We're editing just a very small percentage of it. That may not make a big dent in an odometer metric, but it's going to show up in a big way in a speedometer metric. So that's the logic of the DunedinPACE approach and the reason we focused on this longitudinal rate of change phenotype rather than the cumulative phenotype of mortality risk or years of life lived. I should note, you mentioned this kind of longitudinal design is only possible in a very small number of studies. We've been developing methods to expand the application of pace of aging methods into other cohorts. We, just the other day posted the preprint for an implementation in the Framingham Heart Study offspring cohort. We'd previously published in the U.S. Health and Retirement Study. So it is possible with some additional methodological steps to analyze longitudinal data in other cohorts. But what makes the Dunedin study so special, and I think what allows it to punch so far above its weight class in terms of scientific impact, is just how clean the design is. So it's relatively small cohort, it's 1,000 people, but because they're all born in exactly the same year and all measured in exactly the same way at exactly the same time, it really frees the analysis from many of the biases and sources of noise or error that we have to then deal with statistically in other studies. And so we can think about the longitudinal change phenotype as the crux of the aging process, what we need to measure in order to develop an aging biomarker that can serve as a viable readout in a clinical trial. And the Dunedin study so far is as close to an idealized setting in which to develop one as at least I've encountered.
Yeah, it's quite incredible. And I think we've mentioned a little bit here just in the variety and how many different dozens of biomarkers there are out there. And as we said, some predict age, some predict mortality, some others, disease. But in your view, what should a biomarker demonstrate before we can confidently say that it measures biological aging rather than simply a correlation with age? And if I can just go back into a previous question, we said that the DunedinPACE is up to 45 years old. Have we started seeing aging shifts within the population?
Let me take your last question first, because I think this relates back to questions about the original observation of the pace of aging. And I sort of buried the lead, perhaps, in that the most striking observation to many people about that 2015 paper was that we were observing in the bodies of 26-year-olds over a 12-year follow-up period the same kinds of changes, the same directions of changes that we were used to seeing when we tracked people as they aged from 60 to 80, let's say. So we could see blood pressure go up, signs of inflammation increase, lung function deteriorate, cardiorespiratory fitness decline, kidney function decline, liver function decline. So across the board, we saw these people's bodies changing in the same ways in which we know that they change later in the life course as people descend into disease and disability. These changes were in some cases more subtle than we would see later in life, but they were significant. And critically, they were also variable in this population of young people. So not only were people's bodies changing as we expected them to, there was a wide spectrum of variation in that change. And that allowed us to ask the question, is this change meaningful in terms of what people are able to do in their lives? And what we saw when we asked that question was that people whose bodies were changing more rapidly showed diminished physical and mental capacities at the end of follow-up. So they had poor balance, they had weaker grip strength, they performed less well on tests of neuropsychological function, and in fact, the journalists particularly like this one, they looked older when we showed facial photographs of them to Duke University undergraduates and asked them to rate how young or old people looked. These days, of course, we do all that with AI. But at the time, the state-of-the-art technology was a panel of 20-somethings who were going to rate whether a person looked old or young. In fact, now we're digressing substantially, but sometimes the question was asked, can these young people actually tell the difference between folks who are a couple of decades older than they are? And the answer that we learned was that they're actually the ideal raters. Plastic surgeons have for quite some time been using these young raters to evaluate the efficacy of the interventions that they're delivering. And the theory behind this is that these young people are going to be optimally sensitive to signs of youth and health because those were of evolutionary significance. Now, that's all essentially speculation, but it is kind of an interesting anecdote about how that particular design of the study came about. So I've answered your question about, did we see aging-related change in the Dunedin study participants, and was it meaningful? So the answer is yes and yes. You've asked another question, which is, what makes a good aging biomarker? And when we entered the field, there was a set of criteria that were established, maybe a decade or two earlier, that really focused on whether a molecular measurement was correlated with chronological age, and whether it could be translated into a mouse with fidelity. And we sort of thought, well, neither of those are particularly sensible criteria as we're migrating out of an effort to understand the basic biology of aging and toward a place where we're actually trying to implement measurements in humans in the context of clinical trials. So that's take nothing away from the original conception of those criteria, but simply that they needed to be re-envisioned for our modern geroscience era. And I think that a lot of those details ended up in the Biomarkers of Aging Consortium's consensus report from 2023 that Raghav, I believe you're an author of, as well as myself. The lead author is Madi Mokri. And there, many of the ideas that we introduced back in 2015 about what the right criteria were for an aging biomarker, had sort of reached consensus by that point. And these include, in the first instance, prediction of aging-related morbidity and mortality. More important than whether a biomarker can help me determine whether a person is chronologically old or young is whether that biomarker can differentiate for me among people who are the same chronological age, which ones will enjoy more healthy lifespan going forward and which ones will experience less of that. And so that's really the first and most critical criterion. The second thing that we asked about the pace of aging and that we've continued to ask about other aging biomarkers that we consider is, do they appear sensitive to exposures that are known to shorten healthy lifespan? So looking ahead, we can say, can they predict the things that an aging biomarker ought to predict, how long you're going to live, how much health you're going to have? But also in looking backward, do they accurately reflect a history that either would forecast a shorter healthy lifespan or a longer one? And that's not to say that there isn't opportunity for resilience or recovery, but that on average in a population, a biomarker of aging should indicate older biological age or faster pace of aging in people who have experienced traumas, suffered chronic adversity, have fewer economic resources, experience a lot of stress, things that we know lead to the earlier onset of disease and disability. And then the final criterion, and this is quite a tough one, given where we are in the field today, is that they should be responsive to interventions that modify aging biology. And The trick here is, of course, that we want to use these biomarkers to determine whether interventions modify aging biology. So it's a little bit circular at this stage in the sense that while we have interventions that are theorized to modify the biology of aging and that we know can do that in worms, flies, and mice, we don't yet have validated interventions in people. So what we're leaning on here really is when we translate interventions from the laboratory model space into people, do those interventions lead to changes in these candidate biomarkers of aging? And we have a small but growing number of those kinds of interventions that are being tested. And Raghav, you've of course done work to curate large numbers of those experiments into this translation database, which is a really exciting resource to do this kind of validation.
Yeah, of course. And Dan, I actually really love your framing here, which is around there's one test around responsiveness, right? Which is for the interventions itself, which is kind of our holy grail of sorts. But you're also saying that there needs to be kind of this positive control, right? Which is that you need to be able to show that these biomarkers are accelerated or essentially changing when we know things that do shorten health span and lifespan, such as sort of stress. But to your credit, you have actually shown that these biomarkers, and especially DunedinPACE, is really responsive in interventions that we presumably know are pro longevity interventions, such as calorie restriction, which is the work that you showed in your calorie trial study, which of all these different biomarkers, including sort of grim age and pheno age, DunedinPACE was the only one which was sensitive enough to this change. Why do you think that was the case? I think you already alluded to this a bit earlier, but maybe sort of give a deeper insight into that.
Sure. So, yeah, I was lucky to get to work with the CALERIE trial. This was an effort run by the National Institute on Aging at 3 clinical sites in the United States, at WashU St. Louis, at Tufts, at Pennington Biomedical Research Institute in Louisiana, where they took the evidence from the laboratory models that caloric restriction, so just broad macronutrient reduction with maintenance of what we call micronutrient sufficiency. So it's not starvation, it's not malnutrition, we're just reducing the total caloric intake, has these broad pro-longevity effects in many laboratory models. Not all of them, and there are critiques that suggest that really all we're doing is curing the chronic obesity of the laboratory mouse. But
Yeah.
broadly, we see evidence in caloric restriction experiments in laboratory models that the biology of aging is being modulated in ways that cause this extension of healthy lifespan. And so NIA launched this trial to test whether we could see evidence of the same kinds of processes in people and critically determine whether it was safe and feasible. And so CALERIE 2, which is the trial that produced these data, was the first and remains the only trial of long-term caloric restriction in healthy, non-obese adults. It's a 2-year trial. And we began by looking at blood chemistry data from the trial. We implemented algorithms that had been proposed by Morgan Levine in one case, an implementation of Clemara and Dubal's biological age algorithm, and by Alan Cohen in another case that aims to assay systemic dysregulation, to ask whether the trial showed signs of positively influencing physiology in ways consistent with a delay in aging. And we published that data back in 2017. Shortly thereafter, Bill Krause and his colleagues published another report showing that across the board, calories seem to improve the cardiometabolic health of participants. And across the network of investigators in the trial, there have been a great deal of data published suggesting that caloric restriction downregulates inflammation. It is not dangerous to people in the ways that we were concerned that it might have been. It didn't cause wasting of organs or muscle. People actually tended to feel a little better when they were on the CR diet. They slept okay and so forth. And so the data that disease risk was reduced, that organ integrity and function were maintained, and there was substantial weight loss with stabilization as expected, suggested that this was an intervention that was working more or less in the way it was expected to in laboratory animals. And so with additional support from the National Institute on Aging, we were able to measure DNA methylation from blood samples collected from the participants at baseline, 12 months, and 24 months of follow-up, and then ask if the intervention actually modified the ticking of these different aging clocks. And Raghav, as you said, what we found was that the clocks ticked, So when we looked at chronological age clocks and mortality prediction clocks, they did show people getting older about a year at a time across the 2 years of follow-up in the trial, but there wasn't any difference in that rate of change between treatment and control. In contrast with DunedinPACE, what we saw was that the pace of aging was reduced in the intervention group from baseline to 12 months, and then that slowed pace of aging was maintained through the second year of follow-up in the trial. Now, when you test a bunch of things and one of them stands out and the others don't, the first concern is, of course, that this is just a random chance event.
Yeah.
And so we have some statistical corrections that can rule that out, but those only go so far. So ultimately, the real validation will depend on analysis in a fully independent trial of caloric restriction or a comparably evidenced intervention on the biology of aging. But there was, in fact, another criticism that some of our reviewers raised, which was that the people in the CALERIE trial were on average 38 years old at baseline, about the same age as the Dunedin Study participants whose data were used to develop this DunedinPACE biomarker. And that was in contrast to the people whose data were used to develop the PhenoAge, which is a mortality prediction algorithm, or the GrimAge, which is another mortality prediction algorithm. And we know PhenoAge and GrimAge can predict morbidity and mortality. We know they're responsive to life history exposures in the sense that people who've had harder lives tend to look biologically older. People who are taking better care of themselves tend to look biologically younger. So they have some validation evidence. And it's a little concerning that they're not showing response to this intervention. Maybe it's because the people whose data were used to develop those algorithms were older. And so it's the alignment of the life course stage of the participants in the CALERIE trial with the people who contributed data to develop DunedinPACE that explains the sensitivity of the clock. And we took that criticism seriously. And in our paper, what we argued was we have to acknowledge that possibility. But our argument is that it's not about the people, it's about the method. It's this speedometer-odometer distinction. So the GrimAge, the PhenoAge, they're summarizing the full extent of aging people have accumulated across their lives, in some cases 30, 40 years before the intervention begins. And we're perhaps changing what's happening biologically for the next 2 years with this caloric restriction intervention. But if we're not actually reversing aging, those odometer type clocks may have relatively limited sensitivity to detect the differences that are arising in people's bodies. In contrast, with DunedinPACE, this speedometer type metric, it doesn't really matter how much of the life course you're changing. It matters that you're changing things enough right now that you've meaningfully slowed the pace of aging. But at the time, we couldn't adjudicate between those competing explanations, similarity in the samples or the people who were being analyzed versus the method that was distinct. And that was part of what motivated this effort to develop this new epigenetic clock, Framingham PACE, in data from the Framingham Heart Study. And the motivation there was that these are the very same data that were used to develop the GrimAge. These are people who at the time their DNA was collected to do methylation analysis were in their later 60s and 70s. So these are older folks. Because the Framingham study had accumulated longitudinal data on many physiological indicators, some overlapping with the ones we used in the Dunedin study, most of which actually didn't, but which broadly reflected changes again, in multi-organ system integrity, we could build a pace of aging in the Framingham Heart Study. And then we could model it from this later life DNA methylation to build a second epigenetic speedometer biomarker that we could then put back into CALERIE and ask the question again. And we found we did that was that the Framingham PACE speedometer was slowed between baseline and 12 months, just like DunedinPACE, maintained that slowed pace of aging through 24 months, just like DunedinPACE, and distinct from GrimAge and PhenoAge. So this evidence persuades us, at least, that the differential sensitivity of DunedinPACE relative to these odometer clocks is really about the biomarker design, the method used to extract a pace of aging. And this aligns, by the way, with work that Rich Miller and his colleagues have been doing primarily in laboratory models around what they call aging rate indicators. So hopefully you'll get a chance to speak with him about that work, or perhaps you have already.
He was actually our last podcast guest, so just right about on time.
Yeah. Yeah, exactly. Okay, so this is all incredible work, but there is also new initiatives that you heavily involved in, and one of the biggest ones is the ARPA and the FAST initiatives. which you're leading. For the listeners who might not have heard about it, what is FAST trying to accomplish, and why is this such important initiative in neuroscience?
Yeah, so FAST is now funded by the Advanced Research Projects Agency for Health, or ARPA-H, which is a new federal health funder within Health and Human Services here in the United States. It's part of their PROSPER initiative, which is focused on resilience and aging. And the overall objective of PROSPER is to develop a novel surrogate endpoint for future geroprotector trials and to advance the development of novel therapeutics that target aging or promote healthy longevity. FAST is a tiny piece of that larger picture, but it's focused on an innovative idea that was born within the American Federation for Aging Research, AFAR, under the leadership at the time of Nir Barzilai and Jamie Justice, and then handed off to me when Jamie went to lead X Prize a number of years ago. And the idea behind FAST was that what we're doing to develop aging biomarkers today, or in this case a few years ago, is we're predicting forward. We're trying to model future outcomes, or perhaps in the context of something like DunedinPACE, we're trying to model a concurrent state, how fast people are aging. But these metrics, while they may give us predictive biomarkers, or these approaches, while they may deliver us predictive biomarkers, and we can see GrimAge, PhenoAge, DunedinPACE, these biomarkers are predictive, may not be optimized for responsiveness. In other words, for biomarkers that will be sensitive to the effects of an intervention on the biology of aging. And perhaps we can flip the script a little bit and try to discover these aging biomarkers by analyzing molecules that do and don't respond when intervened on. And so FAST, I think, had this idea, but not necessarily a method for implementation. And as we evolved it through first funding from Impetus Grants and now under the umbrella of ARPA-H, what we have done is attempted to integrate data from clinical trials of multiple FDA-approved interventions that have proof of concept for impact on aging biology. So these are drugs that were curated to show favorable impact on molecular hallmarks of aging, so mechanistic changes in cells related to aging in laboratory animals, and that induce extensions in healthy lifespan in those laboratory animals. So that's the first screen. The second screen is that in human clinical epidemiological studies, these interventions need to protect against all-cause mortality. So not just death from the disease the drug is being delivered to treat, but death from multiple causes. So the effort identified a series of drugs that met those criteria: metformin, rapamycin, GLP-1s, SGLT2 inhibitors, and bisphosphonates, which are drugs that we use to treat osteoporosis. FAST so far has not been able to enroll a bisphosphonate trial. So if you are a drug company and have a bisphosphonate drug with a successful completed clinical trial, we'd love to talk to you about assaying your biospecimens. But what FAST is doing in the case of all of these drugs is we've recruited completed and successful clinical trials that move FDA-approved endpoints. We are now generating multi-omic data from those samples, proteomics, metabolomics, epigenetics, with the goal of testing how each one of those molecules responds to the intervention, and then integrating results across trials of different drugs to find those molecules that are responding in parallel to multiple independent therapeutics. So that's our first requirement in FAST, is that these are responsive molecules. They not only have to respond to multiple interventions in the same way, they have to predict FDA-approved endpoints. They have to predict the incidence of disease, the loss of function, perhaps mortality. I think most of our trials are actually tuned to disease events rather than death, but death is in the mix. Finally, we want to see evidence that they mediate the effects of the drugs on the outcomes. And the idea is that the molecules we identify that meet those criteria will provide an optimal substrate for the development of composites along the lines of DunedinPACE, Framingham-Pace, et cetera. And in fact, there's an independent ARPA-H project, a team led by Mike Snyder at Stanford, that is trying to put together the data we're developing with data they're extracting from observational studies to build a composite that can be implemented in a very inexpensive test kit and then can be handed off to people in yet other ARPA-H projects who are running clinical trials, either repurposing drugs like the ones we're studying in FAST, or who are trying to develop brand new drugs in laboratory models and then get them into humans.
Pretty amazing, sort of the whole ARPA-H initiative in general. It is very powerful. Also, sort of, I feel like you're underselling your work in this, Dan. It's you got a pretty penny to do this kind of work. It's a couple of tens of millions of dollars. So you're not a small part of it at all. You're a big part of it.
We just got 1/10 of million of dollars,
Well,
but
well,
even so, it's a great deal. It's an enormous investment of resources. Our project is meant to be accomplished in just 18 months. So this idea of doing a really big thing really fast to try and catalyze a breakthrough in the field is really powerful for aging research and something we haven't had previously. I think beyond the specific objectives of FAST in terms of discovering these candidate response molecules, and this is something you alluded to in your framing earlier, is that it will build a platform that will allow us for the first time to evaluate across multiple interventions whether aging biomarkers developed outside of our project, biomarkers like DunedinPACE, Framingham-Pace, the Grimm age, some of the proteomic clocks that have been developed, and others are responsive to interventions that we know are acting to extend healthy lifespan, to prevent disease, to prevent disability. And so I think beyond what we do discovery-wise within FAST, FAST is going to create a platform that will allow investigators going forward to rapidly evaluate whether the new biomarker they have is not only predictive, which they can do using the existing cohort data that you and I have analyzed in our own work, but also responsive in the context of a series of different interventions with proof of concept evidence for impact on aging biology.
yeah, I absolutely agree. I will ask you one question on this front, which is, do you expect that one biomarker or one sort of readout would potentially emerge as kind of the clear winner? And this is me asking you, sort of making a prediction here. Or do you think there might be different biomarkers for different purposes altogether? You know, one set of biomarkers that would be really good at predicting disease risk, and a different set of biomarkers for intervention response, and maybe a completely separate biomarker that might combine both of those two to become a surrogate endpoint. Do you see sort of each of them being different biomarkers, or just one biomarker to rule them all?
Yeah, so I think in principle, this idea that you could have distinct response versus predictive biomarkers makes perfect sense. And certainly it may be the case that the biomarkers that we use to evaluate gerotherapeutic interventions need not be the same as the ones we use to stratify people into additional surveillance, potential medical intervention, or even when randomizing within a clinical trial. So I think there's absolutely space for biomarkers that are predictive but not responsive. I think the question of whether you could be responsive but not predictive is more complicated. So certainly you can get away with being less predictive than the most predictive marker if you're going to be a response biomarker. But at the end of the day, because we're trying to infer the significance of these biomarkers for the biology of aging from the way they respond in these interventions, we do need to discipline them with an expectation that they be predictive of health span and lifespan in some capacity. If they're not related to either future health span and lifespan, or at least concurrent physiological and functional integrity, then the question arises, are they really measuring aging, or are they measuring something about the sort of pharmacodynamics or pharmacokinetics of the intervention itself, which might be interesting from the perspective of drug development, but might not be very useful for us, as you're suggesting, in terms of envisioning future surrogate endpoints or even early phase readouts in trials to tell us whether a drug or an intervention is on the right track.
So let's shift a little bit. And I know that these initiatives are sort of like part of this question that I'm going to ask, but in 10 years, looking ahead 10 years, what do you think a success looks like? And what do you hope that the FAST initiatives will change about how aging clinical trials are conducted and how they can be used as biomarkers to evaluate endpoints or therapies? Because I think one of the sort of like what slows down medicine right now is that we come from a place where we're treating the disease. So you're only offering the treatment after you have sort of the disease installed and you have passed that threshold. What we're trying to achieve here is how can we intervene before all of this occurs? And with that comes the risk of, are we giving a treatment that might not be necessarily needed for someone, right? So that's, I think, one of the things that regulators think about before accepting those biomarkers into as a valid endpoint. So do you think that FAST will be able to shift that? And how do you see this happening?
That's certainly the vision of Prosper, is to get us toward a place where we can intervene for prevention. We're comfortable with the idea that we can change health behavior to prevent disease. And I think we're growing to appreciate how important it is to design the environments that people live in ways that contribute to the prevention of disease. But the notion that we might intervene medically in a preventative capacity is new and I think remains, as you point out, uncomfortable to regulators and to, I think, a lot of folks in medicine. I hope that, you said 10 years. 10 years is an awful long time. I feel like AI and the pace of its development have really changed what we're all prepared to speculate about. Well, I think by next week, things are still going to look pretty much the same. 2 months from now, I don't know. You want to talk a year, 5 years, who knows? We could all be on the moon. But I think, from a conservative standpoint, assuming we don't achieve artificial general intelligence, I think my hope would be that by that time, we really are in the field trialing these preventive interventions, that we have succeeded in persuading regulators that there is something actionable in the biology of aging in humans, that there are reproducible, reliably measured molecular changes that occur within and between our cells that contribute to the development of what they already recognize as treatable diseases. And that by intervening on those molecular processes, we can effectively delay or prevent those diseases, granting people more healthy lifespan, granting societies more productive capacity, reducing the burden of healthcare expenditures, at least in the short term, and really changing the way we're modeling our aging human populations from a policy and programming standpoint. So what I'm saying is, I think in 10 years, I want to see us running those clinical trials. I don't know if we'll be there yet. I don't know if we'll have the interventions. I also expect that by that time, we will have reliable and valid aging biomarkers that are routinely available in clinical settings, that our electronic health records will carry embedded information about our biological age and pace of aging. And I think that FAST is a small piece of the data infrastructure necessary for us to get to that reliable and valid biomarker endpoint. We need data from completed clinical trials so we can ask whether these putative biomarkers of aging are responsive to intervention, whether they mediate the effects of intervention on disease. So if this drug modifies aging biology, in some ways it should move the aging biomarker, and disease prevention should be proportional to that movement. And I think that whether the trials that we generate data for within FAST are sufficient to get us there, or simply demonstrate the model for what needs to happen at a larger scale, they are helping to shift the conversation and set a new target for where we need to go. And we're seeing some positive evidence of that already, as some of the big pharmaceutical companies are picking up this idea that there could be something here in targeting aging, and that prevention is a frontier that has the potential to be actionable. And if they will generate these high-dimensional molecular data from the biospecimens they're already collecting in their trials, they may be able to mine those data to devise some of these metrics, or at least to test, validate some of the metrics that are being developed in the research space.
Well, I guess all of that is based on the fulcrum that AI doesn't take over our jobs, Dan, in the next 2 or 3 years.
Well, I mean, if AI takes over our jobs, maybe we'll get it all done faster.
Maybe that is true. And we can all paint and eat and do things other than thinking about aging. Before we let you go, Dan, I want to ask you one final question. And this is a question we ask all our guests. Who is a scientist or clinician or thinker in the longevity space that you'd like to hear on Decoding Longevity? By the way, this question always enables us to have new guests on our podcasts. So I'd love for you to sort of share who you
Yeah,
think.
sure. So, I mean, I think one of the people I would encourage you to talk to is Terry Moffat. So we didn't talk about this in the history of the Dunedin Study, but the original Pace of Aging paper that I wrote with Terry Moffat, I started writing when I was her postdoc, and I finished writing when I was her junior faculty collaborator. So Temi has been a deputy director of the Dunedin Longitudinal Study for many years. She began collaborating with that cohort funded by a K Award, I think back when the participants were adolescents, and has really led the shift of the cohort into the aging space. And it was her vision to accumulate and align these longitudinal data to work toward this idea of a novel measurement of aging. So I think her insights about that and perhaps the many other rate metrics that she and her team are beginning to derive from the Dunedin study would be interesting to hear about. Other people in the aging and longevity space. I mean, I think you probably know these folks at least as well as I do. So I'm trying to think of people that might otherwise not be on your radar. I think that some people who are off the beaten path in geroscience who would nevertheless be interesting to talk to might include Riccardo Marioni in Europe, who's been just developing extraordinary data resources, especially the Generation Scotland cohort. Sarah Haag at Karolinska. She's, fairly well integrated in geroscience at this point, but just thinking Europe-wise, I think she'd be an important person to talk to. There are some junior folks who have interesting ideas about how what we've been doing with aging biology and aging biomarkers may change the way we study even younger people.
Okay.
So Laurel Raffington at Max Planck Human Development in Berlin is one, and Emily Courtin at London School of Economics is another. I won't be pilloried for all of the fantastic people whose names I didn't mention. Again, many of them I think don't need to be brought up by me, but I'll continue to ponder who among my colleagues might be good fits for your show and push them in your direction.
No, that's an amazing list, and we'll definitely have a few of them.
Yeah, thank you, Dan, for this incredible talk and how to share a little bit how incredible your work has been and how it's shifting the thinking from asking how old we are to how quickly are we changing, right? So that seemingly simple shift has profound implications not only for how we study aging, but also for how we design clinical trials, evaluate interventions, and ultimately improve health longevity. So thank you so much for joining on the podcast.
My pleasure to be with you.
Yeah. And thank you everyone for listening in. We link Dan's work around DunedinPACE, the ARPA-H FAST Initiative in the show notes. And if you enjoyed today's conversation, subscribe, leave us a review, and share the episode with someone interested in the science of healthy aging. Until next time, let's keep decoding longevity.